of large RNA 3D structures is today an extremely hard and difficult problem. And in RNAs, large start at around 50 nucleotides, which is not that big. So in the past decades, a lot of people had worked and developed wonderful techniques to predict the secondary structure. Thankfully for me, going from the secondary structure directly to 3D has not been as easy as it seems. At the complete opposite of the spectrum, hundreds of RNA had been determined by crystallography or NMR, and people have developed other techniques to try to break them down into, let's say, motifs or building blocks for larger 3D RNAs. The whole goal of my framework is to try to merge those two opposite sides of the RNA prediction world to try to fill those secondary structures with motifs in 3D and try to ease the final 3D reconstruction. So I will first start by talking quickly about RNA structure, what I understand by motifs, and the structure prediction paradigm I'm going to use. Then I'm going to introduce the framework, some math around it, and finally, results we obtain with all that. So as you all know, RNA is composed of a sequence of nucleotides, AUGC. And what's particularly interesting about RNA is that it has an incredibly flexible backbone. And it is single-stranded, allowing it to have some really, really weird conformations. Also, usually we hear that only GU or AC nucleotides bond together, but in fact, any nucleotide can bond with any other nucleotides. And when we look at the 3D structure, what's interesting is the geometry of those interactions. And as it has been determined by Leontis and Wenstoff in 2001, here is an adenine nucleotide, which has three sides, and any side can bind with any other side. So you have roughly around 12 different conformations for any pair of nucleotides that can bond together. So as soon as you have 50 nucleotides, the number of possibilities is absolutely incredibly huge. So in the meantime, people try to solve an easier problem, which is the secondary structure. Usually, secondary structure only take into account the AU, GC, and GU interaction. Why? Because those are the strongest, or the first one that has been observed. But what's mostly interesting about them is that they are so strong, we've been able to determine exactly the energy of those interactions. As soon as we have energy, the first thing that comes to mind is can we try to predict a structure that minimizes this energy? And in the past decades, algorithms have been developed, really fast algorithms when you don't consider pseudoknots, and they have in the best case around 70% accuracy, which is not that bad. Of course, this is always the best case. It turns more around 60% in a more general prediction. But as you can see here, this is exactly the same RNA in two different views. You have those huge parts in the middle that seem to have nothing interesting inside. But in fact, inside, you have a lot and a lot of interaction, which are a lot more free and can take a lot of more different configuration in space. Thus, probably all the interesting thing about the function of the RNA is hidden into those specific structures. So when you look inside, there have been here, this is the first type of what I'm going to call motifs. They can be classified in the number of strands that interact into each motif. The first type, you only have one strand, which are the classical hairpins. And you can already see that although this seems to be void, those two structures are quite different one from another. And if you go then at the second level, you have what we call interior loops. They compose two strands, and they too can have really strange structures. Up to now, algorithms are able to predict those types of motifs correctly. But when you go further with three-way junctions that have three different strands interacting together in highly complex conformation, then algorithms most of the time fail to predict those structures. And as you can see, this is a huge chunk of the total RNA, and the total 3D conformation is centered around this crucial part inside. What we did is we download around 900 RNA Speedy 3D structure from the PDB. And it's been a software developed by Jalil and Denise in 2008 that automatically used the thread annotation of those PDBs to extract cycles and motifs automatically. And this is really nice because it removes all the bias we could have by manually trying to identify what we see as a motif or not. So we were able to obtain around 36,000 motifs, but what is really, really important is that we have only around 5,000 unique sequences for those motifs. So if we look more at the three-way junction by itself, here we have big view, so as you see, totally empty, but those are two similar sequences that form a three-way junction, but that obtain really totally different 3D structure. So as the Lescott and Vestov paper have shown in 2006, you can have roughly 3 different types of three-way junctions, but nonetheless, they are inside each family really different one from another, and trying to predict them is incredibly, incredibly hard. So the idea of trying to use those motifs to ease the 3D reconstruction of our software at the end seemed like a quite good idea. The 3D prediction workflow that I was going to use is the one by the MC-fold MC-sim pipeline, which is a classical paradigm. You start with only one RNA sequences, and you try to predict secondary structures as a first step. And usually you will have a huge pool of secondary structure. And they can be quite different one from another. Usually you're going to order them by energy, try to pick the minimal free energy, but usually it's not even the right secondary structure. And then using the sequence and the secondary structure as a second step, you predict 3D structures. And again, one secondary structure are going to give you pools of thousands of 3D structure, as much as you want, in fact. So the idea is, could we use 3D motifs which are compatible with the initial sequence to enhance the secondary structure? Can you use it maybe to correct the secondary structures? As I said, around 70% of base pair are well predicted, so that gives around at least a 30% of base pairs which are false positive, and really hinder the reconstruction of the final 3D structure. And finally, can we identify the best secondary structure into this set? Best is a really, really vague word. In general, by best, what I mean is, can we identify the secondary structure that represent as well as possible the 3D shape of the RNA that is going to carry the function that we want to observe? Because RNA are molecules, they're constantly folding and unfolding, and we can say that it has only one 3D structure. But we are interested by the 3D structure that carries the function, so we're going to say that the secondary structure that represents it is going to be the best. Since I'm going to have an integer programming framework, we're going to have an objective function, and then this will be able to be a number, easier for us to determine. So in a nutshell, for me, a motif is compatible if the sequence match with the original sequence. I don't go further. As I said, you can have many, many 3D motifs with exactly the same sequence. That's not a problem. For the final 3D reconstruction, I point to the pool of 3D structure that could fit, and the MC-sim software is by itself trying to find the optimal one. He's going to just try them all and modify them to fit with the total solution. Finally, can we try to remove the base pair that are false positives? And which conformation of this joint secondary structure 3D motif is going to minimize the number of free variables, thus easing the final 3D reconstruction? So the first step in this reconstruction, if I start with only one secondary structure, is this is my input. I have the sequence and the secondary structure. Then at the same time, the software is going to insert motifs. And to do so is going to remove some of the base pairs that could hinder a good insertion of motifs. What I show here in red is the three-way junction. And basically only the arrows only want to show that from a motif which has multiple strands, you can always connect all the strands together without crossing any arcs, which is crucial if you don't want clash in the final reconstruction. But we don't stop there. After that, I scripted the MC-sim, giving him all the motifs database, and we went to the final step, which is a full RNA 3D structure. So to do that, I use integer programming, which is simply a system of linear equation. Variables must be integer, and we have the objective function to minimize or optimize. So we decided to use integer programming for two reasons. First, there was this absolutely beautiful paper in 2011, I think presented at ISMB 2, about IP0, which was integer programming to predict secondary structures with pseudonuts, which is, in its largest class, an NP-complete problem. Really, really hard. So we didn't use it with pseudonuts, but only with non-pseudonutted structures. And the idea between integer programming that really was interesting is that it is highly flexible. All the constraints I did into my framework, they're all heuristics. We don't have energy values for those motifs. We don't have real classification. So it's all heuristics. And having a highly flexible software is the best thing. We want to add new classes of motif. I only write one line with the equation. It's done. Remove something, I comment a line. I don't have to rewrite all the software or the paradigms from scratch. I have basically data and two types of variables in my frameworks. First, we have only motifs, which are strands, which x11, xk11 is the first strand, which everyone is a nucleotide. And what's most interesting, of course, the length of this motif. We have a special variable for every predicted base pair that allows us to remove the base pair from the final secondary structure. And finally, we have a variable that indicates where each component of every motif is inserted or not in specific positions. The objective function that I use is fairly simple. Every time I remove a base pair, I add some small penalty. And every time I insert a motif, I give quite a huge bonus, which is proportional to the square of the length of the motif. There's a really simple reason behind it. When we want to do the final reconstruction with MCC, more pieces they are, harder it is to the reconstruction. So with a weight like this encourages the function to insert the largest motif possible and fewest motifs as possible, thus giving less pieces to do the final reconstruction, and again, easing all the process. So I had a lot, and not that much, maybe 15 constraints, which are classified between constraints for different types of motif or base pairs. I think the most crucial ones is first, when I want to insert any component of a motif, since always the goal is to ease the 3D reconstruction, I want to stack the specific component to a base pair, even allowing some overlap of one or two nucleotides, which really, really ease the final reconstruction. As I said, I don't want any crossing arc. We didn't check for pseudonutted structures. This is really harder for now. It could be a next step. So everyone, when you have, as I said, motif, it's really important to be able to link every strand of the motif without ever crossing any other arc. Finally, we don't want any lonely base pair because this doesn't seem to happen a lot in nature anyway. And finally, maybe a really important thing is that components must be ordered. When RNAs have an order, they start by 5', finish by the 3' end, and when we extracted the motifs from the 3D structures, they came with a specific order. And we saw that trying to inverse the order completely breaks the paradigm of reconstruction of MCCM. So having the order is really a crucial step when inserting the motifs. To benchmark all this, we use a dataset of Lang and Schlick, which was in 2010 computational approaches to 3D structures. It has around 40 RNAs. Of those, 11 were of interest of us because they contain three-way junctions inside, which, as I said, are those that actual software are not able to reconstruct. And there was 24 other RNAs that didn't contain any three-way, four-way, or higher order K-way junction inside, and we used this as a negative control. So the first step, there was never a K-way junction inserted if there was none in the original RNA. So that was quite a good thing. Nonetheless, for the 11 RNAs with three-way junction inside, 2 of those had no homologous three-way junction inside the full database of the PDB, so we couldn't do any reconstruction of them. And the 11 remaining, first, the important thing is how did we select and improve the secondary structure? To the far right, what you can see is the secondary structures were predicted with RNA subduct. The final column is the number of secondary structures that were predicted. For a few of them, it's not that bad. You have 2 or 7. But as you can see, there's one with 243 different secondary structures in a quite narrow range of energy. Which one is the most representative? Really hard to say. The column to the left is the structure that was picked by our framework and identified as the best. They are ordered by energy, so especially in this case, you can see that we picked the 163rd structure inside the set. So it might not have been obvious when looking by hand that so far down the stream you have some good structure. Then we try to quantify how well we were able to remove wrongly predicted base pairs. So to the right, you have the average of true positive prediction on the base pairs over all the generated secondary structures. To the far left, you have in the structure that was selected by my framework, how many base pairs were well predicted, and after we removed some of them, how many of the remainings were well predicted. So as you can see, the average over all of the secondary structures was only 63%. The one we picked, the average was 78, and it increased to 84%, which is quite good. But if we look at the nicest example, of course, 3D2G, which has, as I said, 240 different secondary structures. In average, all those secondary structures had around 40% of true positive. In fact, it was way lower than that for the first 100. The one we picked had 91% of well-predicted base pairs. And after removal, all the remaining base pairs were well-predicted. So that shows that as a first step, inserting the motifs and trying to use their information allow us to pick a good secondary structure, good in the sense as said before, and also the base pairs that are removed are mostly the false positive, which is a good thing. But then when we go to the 3D reconstruction, how does that look? This was the original graph, the 3 lines from the paper by Wang and Schlick in 2010 for Eiffel, Rosetta, and MCSIM. You have the length of the RNAs with the best RMSD that was obtained with the reconstruction. As I said, for me, large sat at around 50 nucleotides, and you have 10 angstrom of RMSD. The black line down here is the prediction used with the motifs that we use with MCSIM after. The dots are for the specific length, what is the average of the set of prediction, and this, of course, the line. As you can see, we have way lower RMSD in general, and even then, the slope in function of the length is way smaller than for others. But this doesn't tell us what is good. Do we have this better result because we selected the good secondary structure, or does the motif have really an importance to ease the reconstruction? So then what we did is that we used MCSIM to redo the reconstruction with 2 different types. In blue, we only used the secondary structures corrected by our framework, and we allowed 48 hours to output at most 1,000 different 3D structures. When we used the motif and added directly the information of the 3D motifs, in green, we only allowed MCSIM to run for half an hour because being fast is crucial today, especially for large structures. As you can see, using the 3D motif gives a significant advantage over MCSIM. What you don't see here is that for pretty much all my prediction with the motif, we reached the bound of 1,000 different 3D structures predicted. For most, when we used only the secondary structure, after 48 hours, we had roughly between 15 and 50 different 3D structures, which are anyway way worse than when using the motifs. And for 2 cases, after 48 hours, there was no final 3D structure. It was just not able to predict anything inside. But when we look at RNAs, what's interesting is the function, and RMSD might not be a good weight or a good way to see if it really reflects what are the interaction between the RNAs. So we look at the interaction between all the different RNAs. We use the definition of interaction by MCSIM, which is even finer grained than the one by Lantis and Westhoff. You have around 18 different conformations for any interaction between 2 nucleotides. It takes into account stacking and a lot of others different interaction. In the mass of blurred triangle here, those are the prediction without the 3D motif explicitly given to the MCCM reconstruction. In the circles here, those are the prediction with the motifs. As you can see, again, the increase in positive predictive value and sensitivity of all the interaction of those motifs is really beneficial to use a motif to do final reconstruction. So, future challenges. As I said, the biggest drawback probably of all those techniques is first, we only use heuristic constraint. Then, all the definition of motif we have is really, really small. As I said, we have hairpins, interior loops, three-way junction. We played with four-way junction. But then we can have other classes of motif that we don't have good mathematical definition yet, but I hope we will have soon, as for pseudonuts, and especially, I hope, long-range interaction. Because being able to express long-range interaction as motif, then having the integer programming framework, it would be really, really easy to just add those new types of motif into the total framework. And finally, I think the worst flaw of all of this is the motif database. We only use motif from RNA 3D structures experimentally verified. So all those motifs that are so fragile or probably so interesting that we're yet not able to see them with those techniques, we are never going to be able to predict them. So to use computational biology to try to mutate those motifs seems like really the next goal in all this work. So thank you very much. I would like especially to thank my supervisor, Jerome Valdez-Paul, the François Major Lab, which developed MCCM, especially Karen Saint-Olge and Marc-Frédéric Blanchette, and the ISCB Grant Fund from the Department of Energy. Thank you very much. Any questions?
Hi. Well, it was a nice talk. Can you comment about long non-coding RNAs?
Sorry, about what?
Long non-coding RNAs. Do you think your approach will work?
Well, for now we were focused in non-coding RNAs. As I said, we were up to a range of 128 nucleotides, which was the biggest. We're trying to expand it, of course. Honestly, I have no idea. I hope it's going to work. We're trying a lot of new things now. But I think really the biggest flaw is the database of motif. If the interesting motif that you're looking for has never been observed, we're never going to be able to track it back. But I think that this is a good way, especially for large RNA, to ease a final reconstruction because we give valuable information.
Yeah, thank you.
Pleasure.
So in your data set, it seems that a lot of them were tRNA sequences, right? Smaller sequences of less than 100 base pairs. So I'm just wondering that how large a sequence can you predict? And also, what about, like, for large sequences, chances are you would see pseudoknots. So would they be able to be predicted also?
Well, for pseudoknots, I think probably the best idea would be to merge it with the IPNOT framework, which is also integer programming. As I said, for RNA's length, we went up to 128 nucleotides. And up to that, the problem is when we want to verify if our predictions are good, we don't have that many other benchmark or RNAs, so it's really, really hard. And I think maybe the trick, if you want to go to another order of magnitudes, would be basically to just cut them in smaller pieces, as it's done now, predict for those smaller pieces, and then do the final reconstructions.
So is there any length limit, like you think, like,
A length limit? I wouldn't try predicting the ribosome 16S right now, but I don't know.
Thank you.
No problem. Yes.
Hi, nice talk. You talked about how this structure prediction is a bit of heuristic art and I'm more familiar with CASP and structure prediction of proteins, which is much more developed. And there you see that the best predictors are usually integrated methods that do many types of methods and minimizations and Monte Carlo and expertise. And I was wondering if you plan or have you up to now done some combinations of your method with other minimizations later and such?
Well, we haven't done anything yet because, as I've seen in the graph on the top left, all the others' methods are, first of all, way awfuler than ours. And anyway, most of the time, they're not even able to predict a 3D structure. So trying to combine them for large structure, it's still a completely new field. There's almost nothing done. There's techniques using motif, again, using Bayesian probabilities, stuff like that is starting to be done. But I think RNAs, in comparison to proteins, they're really more flexible. And one structure, it can have a lot of different configuration. You have ribose switches. You modify one nucleotide, you have a totally, completely different secondary structure, thus 3D structure. I think this is a really, maybe a harder problem, or we were not able to see yet the key for the 3D conformation of RNAs.
Thank you.